Hi, good afternoon, everyone. Welcome to Mayo Clinic. And we are so excited to have you here at Mayo Clinic. And we are very grateful for the time and energy you have invested in the IMPACT program. And the ideas you contribute to the area of bipolar disorder are very important and also promising. So we are so eager to hear your ideas in your presentation. However, I should remind you that each presentation should be no longer than 10 minutes. And after that, we will have 3 minutes for questions. Every question is valuable and helpful and very welcome. So let's just start our oral presentation session, and let's welcome the first speaker. Thank you.
Good afternoon. My name is Alexis Loven.
And my name is Catherine Jiraki.
We are here as representatives of the College of St. Benedict and St. John's University with the accompaniment of our advisor, Dr. Catherine Furness. Today we are going to talk about our hypothesis for non-genetic cause of bipolar disorder. Or maybe not.
So many of us have come into contact with bipolar disorder in one form or another, whether it's been watching the personal struggle of a family member, helping a friend, or just talking to anyone about Minnesota springtime weather. But for those of us who aren't as familiar with the disorder, bipolar is an intensely debilitating disorder that fluctuates between episodes of mania and depression. It affects about 5.5 million Americans. The symptoms of a depressive episode generally include a loss of interest in normal activities, a depressed mood, lethargy, difficulty concentrating, and insomnia, where a manic episode can be characterized by feelings of impulsivity, irritability, and agitation, as well as a reduced appetite. There is evidence linking, but because the presence and severity of these symptoms of the manic and depressive episodes vary so drastically between cases, it's very difficult to diagnose bipolar in an accurate and timely manner. Generally, a diagnosis tends to be reached through a process of elimination or trial and error, ruling out other illnesses before even turning to bipolar disorder. This diagnosis is based on the previously described symptoms that a patient experiences that are outlined in the Diagnostic and Statistical Manual of Mental Disorders. Drugs like Prozac or Paxil can be used to treat the symptoms of the depressive stage, and the symptoms of the manic stage can be addressed by drugs like Risperdal or Valium, but there is no true cure currently in existence. There is evidence that links bipolar disorder to a genetic factor with an estimated heritability of about 85%, but a number of cases lack a genetic predisposition, which prompts further investigation into alternate origins of the disorder.
Okay, our hypothesis for the non-genetic cause of bipolar disorder states that a traumatic experience during juvenile development results in cell death, which activates the inflammatory response in the brain. Following stimulation of the inflammatory process, cytokines are released, disrupting the concentration of dopamine and serotonin. Okay, so first of all, before we delve into the specifics of the mechanism, we will first provide a brief overview of the hypothesis. So as I mentioned, a traumatic experience during juvenile development will cause cell death in regions of the white matter in the brain. Excuse me, I'm going to back up a minute. A traumatic experience during juvenile development causes a nervous system response, which will trigger the inflammatory response. And regions of white matter in the brain will get eaten away, if you will, and the regions of the white matter, apoptotic cells in regions of the white matter will release signaling molecules that will activate microglia within specific regions of the brain. That microglia will release cytokines, specifically tumor necrosis factor alpha and interleukin-1 beta, and these cytokines will modify neuronal cell signaling and cause effects that persist into adulthood. In addition to the pro-inflammatory cytokines being released, these pro-inflammatory cytokines will hyperactivate indolamine 2,3 deoxygenase, and that results in an insufficient amount of tryptophan being put towards the synthesis of serotonin, and with less serotonin, a person may experience depressive episodes of bipolar disorder. Bipolar disorder. In addition to that, the pro-inflammatory cytokines inhibit glutamate synthetase, which causes there to be excess glutamate that will cause more frequent action potentials and an increased amount of dopamine, which will result in manic symptoms of bipolar disorder. So just to recap, the hypothesis can be more thoroughly understood with the assistance of this figure. So a traumatic event during childhood or juvenile development, specifically between the ages of about 3 and 18. It could be abuse, neglect, it can be physical trauma, emotional trauma. It will cause, this stress will cause cell death in regions of white matter throughout the brain, and the apoptotic cells will release signaling molecules that will activate microglial cells to release pro-inflammatory cytokines. And as I mentioned, the cytokines that we have found are TNF-alpha and IL-1 beta. The cytokines will then hyperactivate the indolamine 2,3 deoxygenase, which is responsible for shuttling tryptophan into the kynurenine pathway. And in a healthy individual, the tryptophan within the body, it's an essential amino acid, it will go either towards the synthesis of serotonin, and the other half, for the most part, will go towards the kynurenine pathway. With the hyperactivation of the indolamine 2,3 deoxygenase, more of the tryptophan is being shuttled to the kynurenine pathway versus the synthesis of serotonin. So with this depletion of serotonin, a person may experience the depressive symptoms of bipolar disorder. In addition to hyperactivating the indolamine 2,3 deoxygenase, the pro-inflammatory cytokines also inhibit glutamate synthetase. So then glutamate is not being converted to glutamine, and the excess glutamate will bind to quinolinic acid produced by the kynurenine pathway. That quinolinic acid acts as an NMDA receptor agonist, so the glutamate will bind to the quinolinic acid and open calcium channels, causing an excess amount of calcium coming into the neuron and an increased amount of dopamine release, which would simulate a manic episode of bipolar disorder. In order to test this hypothesis, we proposed that a tissue sample from bipolar model mice can be obtained, a neural tissue sample, and then an ELISA test can be performed, which would consist of the cytokines in the brain, the TNF-alpha and IL-1 beta, would be tagged with their associated antibodies. So then when these cytokines are stained, it would give us a clue as to which regions of the brain are more affected by bipolar disorder. And the second two steps would involve dermal biopsies being harvested from both healthy and bipolar patients. And then these samples will go through microRNA-mediated conversion to neural-like cells, or INLCs. And then a modified spectrophotometric assay can be performed with these INLCs to measure the activity of the indolamine 2,3 deoxygenase. And then that would tell us if that piece of the hypothesis is accurate. And we can also use the INLCs to measure the amounts of dopamine and serotonin within that sample.
So the inflammatory response itself is becoming a lot more important in determining a lot of the pathophysiology of many illnesses. And what we're looking at is the connection that bipolar shares with this response. So the current assessment of bipolar disorder is subject to a number of very flawed procedures, mostly being the trial and error method of determining whether it's bipolar or not. The time between initial onset in this particular method of the trial and error, the initial time between the onset of the disorder and the actual beneficial construction of a treatment plan is significantly delayed. So as it is a successfully tailored treatment plan, it does take time to both construct and implement because of how individually specific the conditions and symptoms are to each patient. And throughout this process of time that it takes, the lives of sufferers are continued to be disrupted, and sometimes it's even impossible for people to carry on living a normal life. Our hypothesis will potentially significantly improve the process of diagnosing and treating bipolar disorder. The presence of the cytokines, like Lexi said, can be confirmed or rejected just on a simple blood test that's collected by taking TNF-alpha and IL-1 beta. With corresponding antibodies, then we can come up with a more accurate diagnosis. This allows for the possibility of a reduced rate of misdiagnosis and an expedited construction of a treatment plan. And in addition to these benefits, the INLCs can be exposed to pro-inflammatory cytokines and then to Enbrel or Humira, which are successful and effective means of suppressing the inflammatory response. The success or failure of this course of treatment can be based on the respective increase in serotonin levels and decrease in dopamine levels. In this hypothesis, there lies the potential to brighten the lives of those affected by bipolar disorder, where before there was only the prospect of endless hospital visits, doctors, and endless cocktails of medications. It gives hope and purpose to those who at present cannot function in daily life. Bipolar disorder will no longer be a disorder that signifies the end of an independent existence. This is more than just a hypothesis. This is hope for millions of sufferers who are unable to advocate for themselves. This is the future of regenerative medicine and psychological health.
We would just like to say thank you to our advisor, Dr. Catherine Furness, and also to Dr. Jen Schaefer, Dr. Barb May, and Dr. Clark Cotton of the St. John's and St. Ben's Department of Biology.
And we'd really like to thank Katie Campbell and the whole Mayo Clinic for giving us this opportunity. It's been quite a learning experience and a challenge, and it's been enjoyable every step of the way.
So now we have 2 minutes for questions.
So you talked about how, in the beginning of your presentation, that childhood trauma leads to a decline in white matter.
Mm-hmm.
So is there a correlation between multiple sclerosis and the onset of bipolar disorder? Because they're relating to deficits and same physiology, essentially, or same declines in white matter. Do you know of any correlation? Do you know if bipolar disorder is more prevalent in adaptation?
We have not made that connection. The area that we took the information pertaining to the white matter from was more of physical trauma to the brain, specifically looking at concussions is where we got that from, so we don't specifically know if there is a connection between those, but that's a great question.
In your model, how would you induce childhood trauma in your rodent model?
Expose them to some type of stressor. That's basically what we mean by childhood trauma, is to induce the stress response. So whether that be in a maze or some type of stressor like that.
Great.
So I just have a question about the stressor kind of in general. So you talked about the white matter being reduced because of a physical trauma, but then you want to induce a stressor, and that would be more of an emotional, mental trauma. Is it both? Is it one or the other?
It could be both. It could be either. It could be one or the other. It's kind of
When
encompassed in the.
When we say trauma, it could be physical, emotional.
I thought your pathway was super interesting. Could you explain how so in bipolar disorder, there's goes between a manic state and a major depressive state. Could you talk about maybe how how those kind of relate to each other, how one leads to the other in terms of the neuronal pathway or the neurotransmitters that you discussed?
Okay, well, I'm going to try and explain this without the picture in front of us. But we had talked about how the hyperactivation of the indolomine 2,3 deoxygenase causes decreased serotonin. So it's connected through the indolomine 2,3 deoxygenase, because then that hyperactivates the kynurenine pathway, and then you get the quinolinic acid and the excess glutamate that will bind to increase the release of dopamine. And there would be a delayed response. So they wouldn't be concurring, but there's a delay from the indolomine 2,3 deoxygenase to the quinolinic acid and downstream.
So kind of piggybacking on that question, yeah, if I can just have a second. So do you propose do you know what pathway is that it leads the microglia to signal the pro-inflammatory cytokines to be released?
The pathway between
The microglia and the cytokines.
once they're activated, the microglia just have to be activated, and then the cytokines are released from the microglia themselves.
Okay, thank you. That's very
So I'm Matthew Dietz.
I'm Brian Hastings.
I'm Janet Jennings.
And I'm Casey Pull, and today we're going to talk to you about the importance of human endogenous retroviruses, a maternal immune response, and their relation to the development of bipolar disorder.
So we didn't really have we had a concept of what BE was, but we really wanted to get into the details so we knew what we were talking about, so we could create a really complete pathway. So what we learned was adolescents are 4 to 7 times more likely to acquire BE if the mother is diagnosed as compared to a paternal diagnosis. It's closely related to schizophrenia. Lithium is actually the most common treatment because it's antiviral. A misdiagnosis is a problem, so usually when kids show like signs of BD, people usually misdiagnose it as ADHD or ADD, and so we'll talk about that a little bit more. And it's characterized by a wide variety of symptoms. As you can see in the picture, you have a depressive state, then you have a manic state, and then you have a depressive state later. And so you can kind of see the brain activity that's going on there.
All right, so for the overview of our project, we will show that an infection can activate HERVs, which is external trigger to HERV activation. These human endogenous retroviruses, once they are activated, are linked to a cytokine imbalance in the body. And then this cytokine imbalance, when paired with increased cortisol levels, are toxic to a fetal brain development. These resulting abnormalities that we see in the fetus are mirrored in people with bipolar disorder. And then cytokine upregulation is also observed during manic episodes, which leads us to believe that this pathway may be active again during every manic episode. But first, what are HERVs? Human endogenous retrovirus is essentially ancient viral DNA that's in every human genome. Our genomes are about 5 to 8% HERVs. Many of them are actually beneficial and used by the body. One is used in healthy placental development, another in trophoblast attachment, and other examples. And some viruses are actually shown to have the ability to activate dormant HERVs. These viruses include herpes simplex virus 1, HIV, and common influenza, among others.
So, as Matthew stated, these viruses are known to activate HERVs. So, HERV activation leads to the production of what would be considered foreign proteins in the body, therefore triggering an immune response. And if this immune response is triggered within the mother, this would cause the increase of IL-2, IL-6, and other specific cytokines, thus causing malfunctioning neurodevelopment within the fetus. Also, because viruses are also associated with fever, we also looked at the implications of high fever and the effects on neurodevelopment as well.
So as we were saying, when it's paired with high cortisol levels, elevated cytokines are toxic to fetal neurodevelopment, and the defects that we see in the fetus's brain are an increased ventricle size, a reduced corpus callosum, and a reduced prefrontal cortex. That was a recent study done just with bipolar patients. And so these cytokines elevated have been tested in a mouse model.
Yeah, so as you can see here, so we have normalized pre-pulse inhibition. And so what's being depicted is the children whose mothers were injected with IL-6, and this statistic is being compared to the control group. And as you can see, there's a statistically significant difference between the pre-pulse inhibition of the control and the mice whose mothers were injected with IL-6, obviously a lower pre-pulse inhibition.
Pre-pulse inhibition is a neurological behavior measure, and so it's sort of indicative of bipolar disorder or of a similar symptom in mice, because we can't ask them what's going on with them.
And so BD onset happens around late adolescence, early adulthood, and so we already have a smaller brain volume coming out of them being a fetus and the mother having the infection and all of that. And so then there's another period of neuronal trimming during puberty so that the brain volume gets even smaller. And so the elevated cytokine levels that we saw in the fetus are also seen during manic episodes. The onset is usually paired with a stressor, so life crises happens, and then there's like the full BD onset begins, so something like going to college, moving away, having those new social interactions create this stress and triggers those cytokines. The HRV pathway may also be relevant to triggering future manic episodes. So again, like every single time this stressor happens, they have the high cytokine levels, thus creating a manic episode. And cytokine levels in depressive episodes haven't been studied as much, so we don't know how that affects that.
Right, and just talking about this figure that's presented up here, HML2 is a specific class of HRVs, and what you can tell from the data that when these two treatment groups of bipolar disorder, these are postmortem brain tissues, what you'll observe is that there is a statistical difference between the relative transcription of the specific HRV between bipolar disorder and the healthy control population.
So overall, not only are these HRVs and these cytokine responses active in the development of bipolar disorder, but also during the episodic symptoms of bipolar disorder. Our experimental hypotheses, what we would like to test, is whether or not bipolar disorder has a specific type of immune response, whether a specific cytokine is responsible for these levels, if it's a combination of different one, if that has any relation to the schizophrenia pathway that we've discussed. And then we would also expect remnants of HERV activation, these proteins that were produced, to be detectable in the cerebrospinal fluid or the blood serum of these individuals with bipolar disorder. We'd also like to compare the differences between manic and depressive episodes and levels of cytokines. Yeah.
So what's significant about this pathway? First, this would explain why lithium and other antipsychotic drugs are so effective, because they all have antiviral properties and have all been shown to lower HERV transcription. It would also explain the large lack of paternal inheritance. It would explain the symptoms timing as well as what Janet was saying before. It would also have potential implications for the diagnosis and misdiagnosis of BD, because if it does have a specific signature, we'd be able to take someone's blood and tell them right away whether they have bipolar disorder or some related illness. Stem cell therapy may also help to alleviate the symptoms, as there have been research studies that have shown that they have been promising in helping with neurodegenerative diseases. So it could be a treatment or maybe even a cure.
Awesome. So main points you should take away from this slideshow. HRVs are likely to be involved in the pathogenesis of bipolar disorder. Evidence for HRVs, there's evidence for HRVs as a continued role in bipolar disorder. And then some longer term studies which would be interesting to do would be comparison with schizophrenia, right? Try and differentiate what cytokines are being increased, what cytokines are being decreased, et cetera, et cetera. Cytokine intensity and symptom severity. More cytokines possibly lead to increased symptom severity. Severity of brain abnormalities and symptom severity, seeing what functionally is different from one brain whether or not if something is different from the other brain, that leads to more severity, right? And then HRV activation before adolescence, that's one thing which was a little bit following our hypothesis because we didn't know whether or not HRVs have been looked at into individuals like before adolescence or before the onset of bipolar disorder. That's something which we would like to see. Special thanks to, oh, sorry.
We'd like to give a special thanks to Gustavus Adolphus College, to our advisor, Dr. Caleb DeLorme, in the psychological department, and Mayo Clinic and the Impact Program for giving us this wonderful opportunity to learn and grow as people and write a paper together.
Yeah,
So, thank you for your attention.
Yeah.
Just leave it here.
So now it's 4 minutes for questions.
Yeah, I was just interested, you mentioned, can't remember if I asked you this already, but you mentioned that you would be interested in measuring the levels of cytokines in patients during manic episodes as well as during depressive episodes. I was just curious how you, or if you've thought about how you may go about recruiting the patients during these episodes.
Yeah, we have already, as far as the research goes, we did find a number of articles that said that there was a problem with recruiting people during the manic or depressive episode because they either didn't want to come in or it would just be hard to get in contact with them during that time. We'd have to consult more with people that have experience doing that, as none of the articles specifically said how they did get those people.
We offered them money.
Good money. Other questions?
Yeah, there's an increase in HERV proteins, I guess, during development, either prenatally. Do you think that has an effect on a specific neurodegeneration observed later in life, or as you get organismal development? What I'm trying to say is, do an increase in HERV proteins, do they target a specific cell type or a specific region that can then explain?
So it's not really the HERVs that are attacking, it's more of an immune response in relation to them. But we did do some research into these GABAergic interneurons and their placement within the brain and how that's also an abnormality that's been observed in bipolar patients and how that can affect their functionality later in life too.
Great, thank you.
Last question.
I had a question. Do you guys have specific family of viruses that may trigger the expression of the HRV?
So there's a couple of different ones.
There's a lot of retroviruses. Almost every retrovirus that we've seen has been shown to do that. And then influenza, as we said, some more common ones, but they haven't ever been a huge study yet as to specifically what they all affect, as they are relatively new.
Yeah, our main focus was influenza because we do see a correlation between mothers with influenza during pregnancy and the development of bipolar disorder with psychotic symptoms, as well as this phenomena of kind of a schedule for bipolar development. So if you are born in the winter months, you are more likely to develop bipolar disorder. This correlates with the second and third trimester. It correlates with flu seasons. So if you're getting flu while you're pregnant, and then you are more likely to develop it.
Thank you.
Thank you.
Thank you.
Hi, my name is Jelena Cece.
I'm Tyler Lang.
I'm Catherine Sprung. We're from Anoka Ramsey Community College, and the title of our proposal was Development of Bipolar Disorder in Fetus Caused by Low Progesterone During Pregnancy, LPS, TLR4, and TRICAT-Mediated Pathway. So bipolar disorder is a brain disorder identified by cyclical disturbances in mood, energy, and activity level. It's clinically characterized by at least one depressive episode and at least one manic episode. It's currently cannot be cured, but there are long-term treatment options such as mood stabilizers and antidepressants. Biological changes occur in bipolar disorder, such as alterations in brain areas that are responsible for mood regulation and cognitive function, such as the hippocampus, amygdala, and prefrontal cortex. And also in bipolar disorder, we see lower levels of serotonin and melatonin. So this figure from Mays et al. Is a good representation of a large portion of our proposal. Individuals with bipolar disorder have higher levels of pro-inflammatory cytokines, and those inflammatory cytokines increase the activation of IDO, which is an enzyme that is a part of the TRICAT pathways. And TRICAT pathways take tryptophan, which is an amino acid, and break it down into TRICATs, or tryptophan catabolites. And by doing so, they are taking tryptophan away from the production of serotonin and melatonin. 5-HT is just another name for serotonin. TRICATs themselves also have neurotoxic effects in the brain, so you might that might be another relation to the neurodegenerative pieces that we see in bipolar disorder as well. If we go back and look at the inflammation that we see in bipolar disorder, we might wonder what might cause that inflammation to be higher in bipolar disorder, and some of the proposed methods include exposure to LPS in your lining of your gut. And LPS stands for lipopolysaccharides. It's on the surface of gram-negative bacteria. And LPS binds in your gut to toll-like receptor 4, which is a receptor on the lining of your intestinal epithelium. And when it binds to that, it triggers an inflammatory response, which also decreases the barrier function of your epithelium, making your gut more leaky or more permeable, which then would allow LPS to leak more into your system, into your bloodstream, and possibly have LPS bind to TLR4 in other regions of your body, not just in your gut, which would then kind of be a positive feedback loop for this TRICAT pathway that takes away tryptophan from serotonin. So our hypothesis was that low progesterone levels during pregnancy will cause bipolar in the fetus via exposure to LPS through the TLR4-mediated inflammatory response and activation of that TRICAT pathway.
So to support our hypothesis, we sought some data. And this chart by Flores Espinosa et al. Was done in amniotic epithelium. A similar trend is found in mice fetal monocytes. So if I attract your direction over here, LPS, so this amniotic epithelium was treated with LPS, and it showed upregulation of the TLR4 receptor. At the same time, though, they did different trials with different levels of P4, which is progesterone, and they found that the progesterone had a protective-like effect in the presence of LPS, downregulating TLR4 expression. Then they also did a test of P4, LPS, and R486, which is P4 antagonist, P4 antagonist. So this was the same study done, but rather than look at the TLR4 expression, it was done to see the expression of tumor necrosis factor alpha and interleukin-6, which are two cytokines that have pro-inflammatory properties. A similar reaction was found with P4 having a protective-like effect again when LPS exposure in the expression in the cytokines. So through all of this, we found that it wasn't just in epithelium or monocytes that we were finding TLR4, but also in different brain cells. So this study was done in astrocytes, and what it showed is it went to relate to the expression of toll-like receptor 4 in the presence of lithium being a commonly prescribed medication for bipolar disorder. So astrocyte expression of toll-like receptor 4 was not increased in the presence of lithium. However, it was increased in LPS exposure. When put lithium and LPS together, we showed that levels of TLR4 expression were almost normal.
And so to go ahead and kind of summarize what we've been talking about, we're talking about low levels of progesterone in the pregnancy of the mother as a protective agent against increased levels of TRL4 then expression, increased levels of LPS binding with TRL4, which then is increased pro-inflammatory cytokines, which then increases the TRICAT pathway, and we have decreased serotonin and melatonin levels. So researching our project, we haven't found a lot of information relating maternal obesity and the low levels of progesterone in utero with bipolar disorder. So we think that this is a really innovative approach, especially when you consider the weight gain or the weight cycling within bipolar disorder. We're also looking at the rapid cycling within the disorder itself from a depressive state to a manic state and the subsequent weight gain and loss for that. We're also finding that maternal obesity, the gestational length and weight of the fetus tends to be higher, and that's also a correlation within bipolar disorder mothers, or mothers who have bipolar disorder. The intestinal epithelium dysfunction is not something that's been addressed as far as bipolar disorder, looking at the interaction with TRL4 and LPS. The significance of this and who could be most treated or most helped by this would be pretty much any mother who has a low level of progesterone. At this point, we're finding that the hormonal levels of different women, especially with their obesity, is affecting neurodevelopment. There was subsequent evidence for maternal obesity and autism. Mother is more likely to give birth to an autistic child if she is obese. Again, we are not trying to just address the genetic aspect, but there is a familiar history of mood disorders. So this could be a possible way to test for a woman who has a family history of mood disorder who is also obese, and then her child might then have this complication. Some treatment possibilities are TRL4 has 2 SNPs that are potentially active for gene therapy. And we're also looking at an engineered 2D cell sheeting for progesterone, increasing the levels of progesterone in the endometrium during pregnancy. And then these are our references. And then we would like to acknowledge Kate Campbell, Regenerative Medicine of Minnesota, Mayo Clinic, our faculty advisor, Julie Redinger, then actually Alan Finnberg, Jane Fox, Dr. David Bond from U of M, and Dr. Paula Holmquist.
Thank you. 3 minutes. Questions? Yes.
Yeah, I have one question. As far as the maternal obesity goes, do you have any way that you're thinking about testing that? Because it's, although we've talked about mice models before, it is quite hard to find a good mice model for bipolar disorder. So do you have you looked into that? I mean, as far as
So as far as maternal obesity, that was just one of the aspects that triggers low progesterone that we kind of looked at. There are other progesterone, low level progesterone diseases that cause it. So what we were thinking is for a mouse model, to expose a mouse model with either the TLR4 receptor or not to different LPS and progesterone levels to see what the effect is in terms of the cognitive development.
I can add a little bit to that too. There are also some models we found that have some physiological symptoms that replicate manic or depressive episodes. So you could also look at exposing them to progesterone or LPS and seeing whether their neurodevelopment is affected as well.
Other question?
Thank you. No, there's a question? Okay.
Thank you very much.
Well, good afternoon, everyone. Thank you so much for staying and coming out to listen to our presentation. At the end of this presentation, we hope to provide a little bit more insight about the epigenetic effects of corticosteroids on asthma as it relates to the development of bipolar disorder. My name is Glenn Morris.
My name is Ashka Joshi.
I'm Thor Worth. And I'm Olivia Stiller. We are all students at the University of Minnesota, Rochester, where we study health sciences. We'd like to begin by extending a special thank you to Dr. Jesse Barnett for being our faculty advisor, and Professor David Haynes for helping us out and being willing to answer all of our questions.
So bipolar disorder is a group of 4 chronic mental illnesses characterized by unusual changes in mood, energy, and activity levels. It often affects an individual's ability to carry out daily tasks and must be managed carefully throughout an individual's life. Often, it can fluctuate between mania, hypomania, and depression. Hypomania is a less severe form of mania, and currently there are about 6.3 million adult Americans that have been diagnosed with bipolar disorder. So this is a diagnostic overview of how DSM-4 diagnoses bipolar disorder. For time's sake, I won't be going through it today.
So to start off with a little bit of an overview of the genetic contributors to bipolar disorder. We realized that our presentation was about non-genetic causes, but up until recently, bipolar is considered to be a completely genetic condition. There was a strong tie, and there still is a strong tie to genetic contributions. But as more research has been done about environmental studies, it's been discovered that there is an environmental factor. So we isolated 2 genes, the ODZ4 gene, which is shown on the top of the figure, and the CACNA1C gene, which is shown on the bottom, are 2 genes which have been linked to bipolar disorder and have also been linked to SNPs, or single nucleotide polymorphisms. So we chose to study asthma as we reviewed through the literature because asthma is an extremely common disorder and has shown comorbidity. But to begin a little bit about asthma, it's been shown as of 2009, 1 in 12 individuals have been clinically diagnosed with asthma, with corticosteroids being one of the most popular recommended medications to treat it, followed by bronchodilators. With the medical expense being about $1,300 or $3,300 per person per year, it can get quite expensive to treat, and due to this comorbidity between asthma and bipolar, which we found 2 distinct studies which showed this comorbidity. One study in the UK inquired about 20 different medical illnesses in conjunction with bipolar disorder, and asthma was one of the most prevalent, with almost 20% of individuals self-reporting a history of clinically diagnosed asthma. The second study showed that 15.9% of individuals self-reported a history of asthma.
So if you're an individual with asthma, like Olivia said, you may either be taking a corticosteroid, such as prednisone, or you might be taking a bronchodilator. Actually, you can take both of them. Both of them work to treat symptoms of asthma. But these 2 types of treatments have been used primarily in Western medicine for about past 16 years. And with this recent medication, there hasn't been a lot of long-term studies on its effect, especially those that relate to mental health as they're trying to treat asthma. So in order to determine the relationship that a corticosteroid might have on bipolar disorder, there's more research that should be conducted. Corticosteroids, like previously mentioned, the figure on your right is the molecular makeup of prednisone. And if corticosteroids were to have any connection with bipolar disorder, a child who has asthma and is taking a corticosteroid now and may develop bipolar disorder later on in his or her life, that could significantly impact the quality of life. That's why we deem it fairly important that it's a relationship that could be better understood. We believe, through our research and some of the other information that we've encountered, that corticosteroids such as prednisone have been associated with DNA methylation, in turn causing different transcription of the DNA, and that is a key factor in the development of bipolar. So with all this information that we have, we hypothesize that the use of corticosteroids to treat asthma causes an epigenetic change due to methylation on the CACNA1C and ODZ4 genes that are common among sufferers of those with bipolar disorder, increasing the chances of an individual who has asthma to be diagnosed with bipolar disorder later on in his or her life.
So the rationale behind the hypothesis that we devised is that we found previous studies showed to have links between the polymorphisms in the CAC-NA1C and the ODZ4 genes and an increased instance of bipolar disorder. And like Olivia mentioned, there's also a comorbidity between individuals diagnosed with asthma and individuals diagnosed with bipolar disorder. So given this information, it is with our intuition that these two genes mentioned above could undergo epigenetic modifications when using corticosteroids, indicating signs of bipolar disorder. So in order to prove or disprove our hypothesis, we propose that a laboratory study should be conducted using the C57BL6 mice with a Th1 transcription factor deletion. This transcription factor deletion is crucial to our study because it has been shown to mimic symptoms of asthma. We would obtain a sample size of about 30 mice and then split them into 3 groups designated as control, test, and placebo. Each mouse will then undergo proteome and transcriptome sequencing, as well as they will undergo a behavioral pattern monitor test to see if they meet the criteria of bipolar disorder. If they do have bipolar disorder, then they will be removed from this study.
As Ashka has already stated, we would be using a population of mice of about 30 mice, and after they are tested using the behavioral pattern monitor, BPM, I'll use that from now on, test to see if they meet the criteria for bipolar disorder. If they do, they will be removed. But if they don't, they will continue on in the experiment, excuse me. And the test is done in a BPM chamber that is a plexiglass whole board floor chamber, as like the one that you see on the right hand side of the screen there, where there are holes in both the floor and all 4 walls. They are all equipped with photobeams that measure their hole poking behavior, and along with this they will be matched with either the criteria that would mean that they have bipolar disorder in the sense. Our mice would be separated into 3 different populations. The control group, which would receive no treatment. The placebo group, which would receive a non-steroid treatment. This could be like the use of bronchodilators, as we have discussed before. And the third group would be corticosteroid treatment. Being that all of, at least for treatment B and C, they are aerosol in nature already for the treatment of asthma, we could just administer the same aerosol to the mice twice a day for a period of 12 months, after which they will undergo the same BPM test to see if they now meet the criteria for bipolar disorder. And they would also undergo a second proteome and transcriptome sequencing. And all of these put together will help us to decipher if our experimental treatment did in fact cause any epigenetic change within the mice. Like I said before, the BPM, behavioral pattern monitor test, would be done again, and everything, including that and the epigenome of the mice, will be sequenced to see if there are any variants within the mice that are left in the study after the 12-month period has been ended. Just is try and establish a comorbidity between bipolar disorder and asthma, as has already been alluded to in a couple of other studies. Right now, the use of genomic sequencing and proteomic and transcriptome sequencing on humans is a little not feasible. So we are recommending this on mice for the purposes of our study. And in the future, we do recommend a transition into human studies once they become more readily available for the different sequencing. And after that, we recommend a multi-year longitudinal study to see even more specifically with humans if there is any more non-genetic causes of bipolar disorder, not just the use of corticosteroids.
So as Thor talked about, the further studies that we suggest is an observational longitudinal study once the information about human genomics and using a variety of different sequencing methods is more advanced. So the observational long-term study would be used to identify a population of children that have been diagnosed with asthma and are currently being treated with or had been treated in the past with corticosteroids. A genome-wide association study would first occur, and then to see if they have the genetic variations which indicate bipolar disorder. If they do not have these variations, they would be kept in the study. If they had these variations, they'd be removed with periodic check-ins, and more another GWS would be conducted. The basic premise of this study, should there be positive results from the mouse study, is to monitor whether an individual's genetic makeup is modified by taking prednisone and whether or not they're diagnosed with bipolar disorder.
I was just curious, maybe I missed it, but are there studies that have examined the effect of corticosteroid inhalers on DNA methylation?
None that we have found. Being that they still are relatively new, at least to us being as young as we are, they are still relatively new. The only studies that are out there that we have found are just their use as the treatment for asthma and nothing along the lines of what they do to epigenetic changes or anything.
As asthma rates have climbed within the population, would you also then suppose that bipolar disorder rates will similarly rise?
That's one reason why we suggested this hypothesis, due to those increasing rates of asthma. We want to make sure that we're not basically, should this hypothesis be true, that could be a serious issue if by treating people with a highly effective drug, we were possibly giving them bipolar disorder. So that's something that would definitely want to be figured out now versus 10 or 15 years down the line when we have a population of half the population or all the population of asthma sufferers that were treated with corticosteroids having this condition.
Last question.
So besides the comorbidity or correlation between asthma and bipolar disorder, what leads you to believe that corticosteroids would be causing this methylation?
A lot of our research was done due to the relative new nature of epigenetics, a lot of the information coming out very recently, and with the number of patients that are being treated with corticosteroids being on the rise due to the number of people that have asthma, there have been more studies coming out about the comorbidity, and because the number of patients treated with corticosteroids is on the rise, that's where we drew the hypothesis from, was because there's more patients being treated with this is on the rise, and this comorbidity is on the rise, then maybe there might be a connection here as well.
Thank you.
Mary Vang, and this is Samantha Storston and John Fan, and we're from Century College. We're pleased to be here today to present to you our hypothesis on the non-genetic cause of bipolar disorder. Bipolar disorder is one of the top leading causes of disability in the world. The symptoms of bipolar disorder range in severity and can affect normal and everyday functioning, disturbing relationships and performance in work and in school. A problematic area is the high rates of discontinuation of treatment, which can worsen symptoms. Reports of induced mania by antidepressants is also a concern. Today, it's estimated that genetics accounts for 60 to 85% of the risk of developing bipolar disorder. Interestingly, individuals who have genetic predisposition to bipolar disorder do not always develop it, implicating that there are non-genetic factors at work. Our research brings light to a disorder that has been well studied but not completely understood.
Using findings from years of previous research, we propose a hypothesis for the non-genetic underlying cause of bipolar disorder. We take into consideration the age of onset being a key indicator for pathogenesis during neurogenesis. Diagnosing young children with bipolar disorder is a difficult, complicated process. Despite that, it was found that childhood or adolescent onset of bipolar disorder was 61% in the United States. With this in mind, we began to look at what types of environmental factors may cause neurological disorders in children. A study using childhood trauma questionnaire results showed that the severity of symptoms in bipolar disorder is associated with childhood trauma, and that further childhood abuse, such as physical, sexual, emotional, and/or neglect, were significantly associated with accelerating staging process of bipolar. A convergent study looking at the epidemiological and neurobiological evidence in the Adverse Childhood Experiences study found a positive correlation between exposure of the developing brain to stress with resulting impairments in multiple structures. The three regions of the brain that were shown to be most impacted by stress were the prefrontal cortex, the hippocampus, and the amygdala. This connection is noteworthy because the aforementioned regions are known to have anatomical structural abnormalities as a result of stress. One such possible abnormality is the presence of white matter hyperintensities. White matter hyperintensities are seen in the MRIs of patients who have neuronal damage to brain tissue, such as individuals who have undergone a stroke. Studies have found that bipolar adolescents have a significant increase in white matter hyperintensities compared to both healthy and schizophrenic individuals. A 2009 meta-analysis that reviewed 27 articles highlighted an increased incidence of white matter hyperintensities in children and adolescents with bipolar disorder. Another study looked at the prevalence, severity, and location of these plaques. They shared the similar result of bipolar patients having higher prevalence compared to schizophrenic patients. In addition, they found that the frontal lobes were the predominant location of white matter hyperintensities. Although we're focusing on white matter hyperintensities in children, it's necessary to acknowledge that they are also seen in adult patients, and that more severe white matter hyperintensities are associated with a greater number of hospitalizations and poor response to treatment. Despite much of the research on bipolar being conducted on adults who have a confirmed diagnosis and have already been treated, we use this evidence as a reason to approach bipolar disorder as a neurodevelopmental disorder that can be observed before symptoms meet DSM criteria. We propose that bipolar disorder is due to the dysfunction of glutamatergic transmission, leading to neurodegeneration caused by stress during neurodevelopment. And now John will discuss the glutamatergic system.
Glutamate is the most prominent excitatory neurotransmitter in the brain. And most importantly, it's used for signaling the amygdala and other brain regions responsible for stress response. Because extracellular glutamate can be toxic at high concentrations, it's tightly controlled in what's known as the glutamate-glutamine cycle. Excitatory amino acid transporters, or EAATs, are responsible for the removal of extracellular glutamate from extracellular space. A study looking at the expression of EAATs in people with mood disorders detected decreased expression of EAAT 3 and 4 in the striatum of bipolar patients. A lack of these glutamate transporters can indicate an increased concentration of extracellular glutamate. In addition, it is thought that long-term potentiation increases the surface expression of glutamate receptors. With the implication that additional receptors are present and there's an abundance of extracellular glutamate, we suggest a possible mechanism to explain the manic and depressive episodes seen in bipolar patients. We do so by examining the functions of glutamate receptors. The receptors we're referring to are NMDA and AMPA, two of glutamate's ionotropic receptors. We're proposing that mania is a result of long-term potentiation resulting from long-lasting synaptic signaling. This is because with an abundance of extracellular glutamate, due to the dysfunction of glutamatergic transmission, there's an overexpression of receptors which leads to an increase in signaling cascades. We suggest that the elevated mood and hyperactivity associated with manic episodes are explained as being a result of this elevated signaling, and that depressive episodes occur when the body is trying to return to homeostasis by inducing a process called long-term synaptic depression. We have found evidence that supports this theory. First of all, studies show that there is an increased expression of glutamate receptors during mania. And anti-manic medications such as lithium and valproate have both shown to inhibit NMDA receptors, which would lessen signaling. Another study showed that antidepressants increase the surface expression of AMPA receptors, which would increase signaling and counter the effects of long-term synaptic depression. These findings support our idea that manic and depressive episodes are related to the amount and activity of glutamate receptors. With these findings in consideration, further studies should look at the relationship between extracellular glutamate and both long-term potentiation and long-term synaptic depression. They should also look into how extracellular glutamate is further implicated in bipolar disorder, a possible direction being to examine white hyperintensities as a result of glutamate toxicity. Glutamate toxicity is when there is a harmful concentration of calcium within the neuron. When a glutamate receptor becomes sensitive, less glutamate is required to excite the cell. Overexcitation can lead to long-term potentiation, which as a result increases the amount of calcium entering the neuron, which then leads to neuronal death. An individual who is overexposed to adolescent distress could develop neurodegeneration from enhanced glutamate levels. The resulting neuronal death from glutamate toxicity may interrupt signaling among regions of the brain, explaining the various outcomes and range of bipolar symptoms. If this is occurring during brain development, it is possible that these abnormalities are causing the interferences that manifest bipolar symptoms. In addition, the resulting death of neurons in brain tissue can be the reason we see white matter hyperintensities on the MRIs of bipolar patients. Although white matter hyperintensities are not the only possible result of extracellular glutamate, they're significant because they can be detected on MRIs, and that could possibly assist in diagnostics. Further studies should explore how other possible results of extracellular glutamate can explain more specific symptoms of bipolar. Now Sam will tell us why what we've been talking about is significant about what we can do with information to further bipolar research.
If our proposal holds true and the symptoms of bipolar disorder are caused by extracellular glutamate leading to these plaques, we propose the use of induced pluripotent stem cells to repair the damage from extracellular glutamate. Transplantations of induced pluripotent cells into ischemic human brains has not been researched. However, mice stroke models have been promising when iPS cells were transplanted into the ipsilateral striatum and cortex. Although it has been previously noted that bipolar disorder brains have white matter hyperintensities, to our knowledge, no research has sought out to treat it in bipolar patients. For years, white matter hyperintensities were overlooked because it was difficult to tell them apart from resulting from other causes. However, significant amount of white hyperintensity in children and adolescents who have not been treated by medications give this physical feature. Therefore, this idea is unique in the context of treating neuronal damage caused by excess glutamate in bipolar brains with iPS cells. If our hypothesis holds true, diagnosis and treatment of bipolar disorder would move the focus from genetics and pharmaceuticals to early prevention in children and adolescents who already show early symptoms such as white matter hyperintensities. Considering predisposition and looking for hyperintensities is significant for families who are looking to prevent or lessen onset symptoms in children and adolescents. Early treatment in children can transform the prognosis of bipolar disorder as it has the potential to allow the brain to develop without decreased disruption from this disease. For children, this can prevent further damage from the disease that would otherwise worsen their symptoms into adulthood. Our hypothesis also implies a new route of treatment for adults, especially those who have severe side effects or little response to treatment. For adults, this innovation has the potential to give normal functioning to patients' lives, allowing them to hold stable relationships and firmly establishing long-term jobs. We would also like to take this opportunity to thank Mayo Clinic for letting us be here and for all of you for listening to us, and especially our faculty advisor, Jessica Bell, along with all other professors who helped us along this way. Without them, we would not be where we are. Thank you, everyone.
first group presented talked about how there's a depression in the amount of white matter. I think that's interesting with the white matter hyperintensities. To your knowledge, do you know if outside that particular brain region, if there's a general depression in the amount of white matter, and it may concentrate specifically in, I think, the prefrontal cortex you guys showed?
When we say white matter hyperintensities, we really mean white spots on MRI. I think that, if I recall correctly, when the first group talked about white matter, they were talking about the white part in the brain where the axons are, the myelinated axons. But we did look at decreased glial density in bipolar disorder, and we did in brains of bipolar disorder, and we did see that, and that could be that connection there that you're talking about. Mm-hmm.
So correct me if I'm wrong, but I think iPS transplantation has been used for Parkinson's models. I could be wrong about that. My question is, before you go doing that, and who may or may not have bipolar disorder, have you come up with a less invasive procedure to test your hypothesis?
We have.
Before you put iPS cells?
Yes, we have. We've discussed and we've talked about it, and we think that the best way to prove if our hypothesis is right or not, or along the right track, is to just look at glutamate levels in bipolar, euthymic, or depressive individuals by using the NMR or the NMR that we just we checked out today during the tour. Yeah.
I was just curious if you could comment a little bit more about how early childhood trauma could alter expression of the EAATs.
Well, we didn't make a connection between childhood trauma and EAATs. We assume that it has to do with the genetic cause of bipolar disorder, or maybe a genetic predisposition, but we tried our best to stay out of the gene area of what causes bipolar disorder. Yeah.
Thank you.
Thank you.
Okay, so my name is Anant Naik. I'm from the University of Minnesota, and I'm studying biomedical engineering. Just as a brief mention, my partner fell sick just yesterday, so he won't be here to join us, but his name is Rohit Murakonda. He's also my classmate in biomedical engineering. So the fundamentals of bipolar disorder. So this is a stat that we've covered a lot in today's presentation. 2.6% of the adults in the United States have been diagnosed with bipolar disorder, which is equivalent to 5.7 million people. So this is a macro level life progression of bipolar disorder, also something that we've talked about a lot. So as we can see, there's transient oscillations in stages of hypomania, mania, major depression, and ultimately resulting in a mixed state towards the end of a patient's life. Now, this is very subjective to how long a patient actually lives, because unfortunately, in times of major depression, patients are often likely to commit suicide. So this is a problem that we're seeing, but ultimately, this is a generic life progression, as shown by a review done by Dr. Grand et al. This is a micro-level background. So specifically when looking at bipolar disorder, we chose to look at a specific region in the dorsolateral prefrontal cortex to give a brief insight as to where that is. You can see the green slice. That green slice is corresponding to the slice that is visible on the leftmost panel. So specifically, we're looking at the region of interest called agranular regions 24. In a study by Dr. Ongur et al. At Harvard University in 1998, he found that in this region of the brain, the glia present in this region were incredibly depressed. So as you can see in familial bipolar disorder and other bipolar disorder, they're depressed, they're lower than the control values. And so we really use this as a mechanism to characterize the development of bipolar disorder. Our idea was if a patient has this part of the brain examined and it corresponds within this level of glial density in this part of the brain, it would help to point us in the direction of bipolar disorder. So the relationship between stress and anxiety. So really what led us to pose the question was, after looking at Iraq and Afghanistan veterans, we found that they were definitely more likely to have bipolar disorder and anxiety mood disorders. A study by the Congressional Research Service in 2013 looked at the VA, and looked at the number of patients that were enrolling, and they found that around 15 to 16% of these patients were coming out with bipolar disorder. So what this really drove us to ask was, is bipolar disorder abnormally prevalent in patients exposed to situations of chronic stress? And the answer that we found was absolutely. Patients that have been exposed to childhood trauma, sexual and physical abuse, and the combat veterans, as we mentioned earlier, they're all exhibiting higher rates of bipolar disorder. So this isn't just a confound that we're experiencing with veterans. This is across the board in patients that have been exposed to chronic stress. So as a characteristic figure, Dr. Otto et al., also at Harvard University, did a meta-analysis looking at multiple studies that showed that a lot of patients with bipolar disorder also have post-traumatic stress disorder. Now this is important because if a patient has post-traumatic stress disorder, they've been exposed to chronic stress, they're living the symptoms of that, and as a consequence we can use that. The comorbidity of these two diseases points to an insight that stress and bipolar disorder have a strong relation, and it's worth investigating that idea. So this is the significance generally. So total bipolar disorder patients, some are given the appropriate diagnosis if they have a familial history that's favorable and the behavioral analysis that's done. But a lot of the times, if they don't have a familial history, they have the behavior, it results in a misdiagnosis. So essentially, by including chronic stress history in our inclusion-exclusion criteria, we can effectively select for people with bipolar disorder, giving them the treatment that they need. So our hypothesis is that chronic stress results in an upregulation in endocrine response. So as a result of this endocrine response of the adrenal gland, it releases glucocorticoids, and as a consequence of that, cells in the prefrontal cortex degenerate by interacting with the with these glucocorticoids. So as another kind of visual, as you can see, glucocorticoids before the astrocyte engages with them, and then they come into contact with the astrocyte receptors bind to the glucocorticoid receptors bind to the glucocorticoids, and that leads to the development of bipolar disorder. So our rationale for this really comes from a series of studies. The first one was looking at in vitro cell cultures. They looked at cortisone as a glucocorticoid and applied it to a cell culture of astrocytes. And by this application, they saw that the increased concentration of cortisone led to a decrease in the amount of functionality of these astrocytes. This was done through an MTT assay. Second thing they looked at was the percentage of BRDU positive and GFAP positive cells. So BRDU is a marker that measures for cell proliferation, but also GFAP is just a general measurement for astrocytes. So this also decreases, so the number of astrocytes themselves are declining. And finally, cortisone impact on gRNA prevalence was also there. So siRNAs, or small interfering RNAs, essentially prevent the expression of the glucocorticoid, the receptor in the cell. So if we decrease the amount of RNAs that are preventing this expression, the number of receptors increase in the cell, which also is correlated with higher likelihood of cell death. Then we also looked at in vivo. So the same study, Dr. Unimera et al., also looked at injecting ACTH, which is the connection between the pituitary gland and the adrenal gland. This hormone essentially causes this upregulation in glucocorticoids. So the increase in ACTH, by injecting it into mice, we essentially saw a decline in the number of astrocytes, as shown by the decreased cell density of GFAP. This is also quantified in the second panel on the right, which shows that aggregate astrocytes for the medial prefrontal cortex and hippocampus were significantly reduced. Now, we didn't look at the hippocampus specifically in our initial analysis because that's not what Dr. Ongur at Harvard looked at. He simply looked at the prefrontal cortex, but in the future, I think it is important to look at the hippocampus. Right now, based on our current criteria, we're just looking at the prefrontal cortex. This also led to a decline in the volume of cells. So the volume of neurons. So a study done by Dr. Serkira et al. In 2007 found that with pre and post-stress analysis, they saw a decline in the volume, specifically in layers 1 and layer 2 of the motor cortex, of the prefrontal cortex. This decline makes perfect sense because if the astrocytes degenerate, it means that the neurons no longer have the metabolic function or the framework, which leads to degeneration in neurons. Now the neurons don't completely die off, they degenerate, which is why we only see a decrease in the volume of the cells. They shrivel and then ultimately it's possible that they die, but they don't. This specifically wasn't studied in this study. So the importance of this is that, again, we ultimately, by showing that chronic stress plays a direct factor in the development of bipolar disorder, we can help include it into the inclusion-exclusion criteria when we're determining who has bipolar disorder and who doesn't. And by doing this, we can give people with chronic stress history, but not necessarily the family pedigree that shows the prevalence of bipolar disorder, the appropriate need by giving them things like lithium or the other treatments that have been discussed. These are generally the experiments that we propose. First, it's important to verify that the amount of expression that is prevalent in bipolar disorder, as shown by Dr. Ongur, is also held consistent with the other studies that we showed just recently. So we would measure decline in glial densities by exposure to chronic stress in naive mice. This would likely be done by injecting ACTH into the mice directly. Second, we would conduct behavioral tests, which would help correlate the decline in glial density to the behavior and the progression of bipolar disorder. And so this would help us to determine when the mouse is exposed to mania or depression. Finally, we could try blocking glucocorticoid receptors on astrocytes using just general blockers, which would help us to determine whether or not this prevents the onset of bipolar disorder in mice. And finally, we want to try stem cell therapy. Now, stem cell therapy would be a last test. We could promote astrocyte growth in the prefrontal cortex by using stem cell therapy in naive and bipolar mouse models. Now, in the past, as we talked about before, it's been shown to work in Parkinson's mouse models where you can inject stem cells in dopaminergic neurons into the striatum, and this helps to alleviate the condition of Parkinson's. So we're hoping that if all these different experiments work, we can ultimately show that the stem cell therapy would be a viable treatment for bipolar disorder patients. Just as a brief acknowledgements, I would like to thank Dr. Atsushi Akura, who's in the Department of Neurology, who helped us, and Dr. Russell Carter, who's in the Department of Neuroscience, who is also my postdoc. He's been a wonderful help, and also thank you to Mayo and their impact program.
I was just curious if you could comment a little bit about the role of this deficit in astrocyte number in the actual cause of bipolar disorder, rather than just being maybe a consequence of depressive episodes, for example. Because in your one of your first couple slides, you showed that I think people with major depressive disorder also had a decline in astrocyte number.
Okay, so you're talking about the Ongur et al. Study?
Yeah. Yeah, there you go. Sure. Yeah, so they're the major depressive disorder. They also have a reduction in the number of glial cells.
Right. So that's definitely true. And we can also see why this is true. I mean, bipolar disorder patients also go through periods of extreme depression and mania. I think it's important that we have to, I mean, ultimately when we do the study, we have to control for this fact. So we would have to take depressive mouse models and compare their behavior to bipolar disorder mouse models, because bipolar disorder, as we talked about, our ability to diagnose it has to go through one phase of mania and depression, and so we would ultimately have to control for that variable, because that is very true, that there is a decreased glial deficit in that region of the brain.
I was wondering how you would correlate this low levels of glial cells to the cyclical nature of bipolar disorder, because how I'm understanding it right now is like I can see how that would affect the depressive state of bipolar disorder, but how would it, what it would look like in a manic
So
scenario?
the upregulation of glucocorticoids, right now, what we're focusing on is the fact that these neuronal networks that many people have talked about before are being disrupted. And that's the main point of this. And the disruption, the specific mechanism obviously is not known. And we don't necessarily know how the glucocorticoid receptors are directly influencing any sort of proteins that are being expressed. And so while that hypothesis is valuable, I think it's more important that we hypothesize the fundamental first, and then we can dig deeper. But right now, based on what we know, we can only go so far as to just making a hypothesis about how to induce bipolar disorder in mice, or just the basic level of our understanding.
Okay, thank you. Good evening, ladies and gentlemen. We are students from the University of Minnesota, Rochester, and today we will be presenting on vitamin D and bipolar disorder. My name is Leila Jean.
I'm Katrina Diaz.
I'm Ramya Kunduru.
And I'm Nazla Khani. Bipolar disorder is also known as manic depression, is a neurological disorder that affects more than 5 million adults in the United States alone. And the neurological disorder is characterized by extreme mania and extreme depression. And mania, symptoms of mania may include extreme self-confidence or a sudden energy in mental activity. And then depression symptoms may vary from feelings of sadness to feelings of hopelessness and loss of interest. There are two types of categories of bipolar, namely type 1 and type 2. The two distinct differences between type 1 and type 2, firstly, is that type 1 of bipolar, you experience a mixture of depression and mania onset at the same time. And then secondly, the difference between by type 1 and type 2 is that type 1 you present with mania, and type 2 you present with hypomania, which is a lesser degree or lesser severity of mania. And family history of bipolar disorder increases your likelihood of having the disorder yourself. In a research performed by Steve Bursert, they found that in twins, if one identical twin had bipolar disorder, the likelihood of the other twin having bipolar disorder is up 70% and 25% in non-identical twins. And then with parents, if one parent had bipolar disorder, the likelihood of their child having bipolar disorder is up 30% and 75% if both parents had bipolar disorder.
So our main focus for today is basically about vitamin D. So what is vitamin D? Vitamin D is basically a fat-soluble vitamin that can be found in many dietary sources as well as sunlight exposure. So the major role of vitamin D in the body is basically to maintain blood levels of calcium and phosphorus, but it has also been found to play a significant role in preventing and treating neurological diseases, including but are not limited to Parkinson's disease, seasonal affective disorder, and multiple sclerosis. In a study conducted by Dr. Ellen Mowry et al. From the Johns Hopkins School of Medicine, they have found that a decrease of vitamin D levels in the body correlates to a reduction of gray matter volume and vice versa. So you may ask, why is this important? Well, another study conducted by Dr. Kiki Chang from Stanford University Medical Center have shown that there is a decrease in brain volume found in patients with bipolar disorder, which kind of like leads us to our main hypothesis.
So putting all of these pieces of information together, we hypothesized that increased level of vitamin D can decrease the symptoms of bipolar disorder. And here's some picture that can visually explain our hypothesis. So there, as we've mentioned earlier, there are multiple ways in which we can obtain vitamin D in our body. And so, for example, sunlight can be one of the sources of vitamin D. And so low sunlight exposure can decrease the level of vitamin D in the body, and which could decrease the gray matter volume. And low brain gray matter volume is observed to be associated with bipolar disorder. So going into details of why our focus
is very important, or why vitamin D is more important in our topic. We found out that 25-hydroxyvitamin D is the active form of vitamin D in the human body. And in a study or research conducted by Dr. Ellen Maury at the Johns Hopkins School of Medicine, she found out that with an increase of 25 nanomole per liter in this kind of vitamin D, there is an increase of 7.8 milliliters of normalized gray matter volume in a person, specifically in the amygdala region. And so we were focused on amygdala region, and we tried to do some research on why this, and we figured out that the amygdala region is responsible for regulation of mood, and it also helps a person perceive and feel emotions in themselves and in others. It's also responsible for behavioral changes. So now, like, going back into the relationship between the gray matter volume and bipolar disorder, we found out in a study conducted by Dr. Williams that the gray matter volume, which is highlighted by the yellow region in the brain, is decreasing significantly in the bipolar disorder patients as compared to the control individuals. And in a study conducted by Kiki Shank at the Stanford School of Medicine, we have found that the individuals with bipolar disorder who are not exposed to lithium, which is a form of antidepressant, have very low amygdala gray matter volume as compared to individuals with bipolar disorder who are exposed to lithium and the control groups. So how do we collect data on this? We figured out that we could do MRI scans on individuals to get the gray matter volume, and we could also do psychological and clinical assessments, such as structured clinical interview for DSM-4 according to the guidelines, and we could do mood disorder questionnaire and general behavior inventory.
So bipolar disorder is a growing psychological condition worldwide. In 2012, United States had the highest rate at 4.4%, with most of the diagnosis being made among young people. Multiple factors need to be taken into consideration when searching for a cause of a condition. So the psychosocial biomedical model is it offers a necessary holistic approach towards health. In our research, we focused on the biological aspect, in particular to vitamin D. There are multiple treatments for vitamin D, and one such treatment is phototherapy. Phototherapy treatment has been conducted under various contexts, such as seasonal depression and jaundice. As stated previously, vitamin D is important is essential for our body function. The National Institute of Health recommends 800 international units for all people ages 1 to 70 years old. For experimentation, our proposed method is to divide all participants into 4 groups. All the participants will undergo MRI scans, as that is the most optimal way to obtain gray matter volume data for our focus region. Psychological and clinical assessments will also be administered, specifically the structured clinical interview for DSM-4 and mood disorder questionnaire. We will also track symptomology, specifically insomnia and concentration, which you can't see on the slide. For example, for insomnia, we will have participants log their hours of sleep, and for concentration, they'll be given challenging activities like puzzles. If our methods are successful, then it can transform the future, with physicians being given a new avenue to diagnose patients by examining brain volume and vitamin D levels. Patients will be given an option for non-invasive and cost-effective treatments with light therapy and supplements. However, bipolar disorder is a lifelong condition, but if the proposed method is effective, it could potentially delay onset of the condition or change length and severity of episodes of mania and depressive episodes, or it could change specific symptomology, specifically insomnia and concentration.
So some ethical considerations, just like any other research, our main proposed methodology also has some risks to it, but our risks are very minimal enough and preventable enough that the benefits actually generally outweighs the main risks of it, because our risk might be overdose, lamp burns, and allergic reactions, which is basically generally non-invasive and very preventable and treatable enough.
If our proposed methods are successful, we have a power to treat or control or transform the diagnosis and the course of treatment of bipolar disorder. Ultimately, the benefit obviously outweighs the risks that are associated with the study, and I think at the end of the day, we would regret the chances that we didn't take. And at this point, we would like to thank all of you for being patient and listening to our presentation, and we want to convey special thanks to Professor James Winship for supporting us and helping us throughout our process of IMPACT, and we would like to thank the Mayo Clinic and IMPACT for giving us such a great opportunity. Thank you. Any questions?
Have you come across any perhaps longitudinal populational studies? For instance, is there a greater incidence of bipolar disorder in northern climates like Minnesota, for instance, versus Texas?
I would say we didn't look into it, but I think it's a good point in context of what we were doing. But I think our focus was vitamin D, but not specifically sunlight, because we were trying to, I guess, convey vitamin D in terms of many other sources out there.
I was just curious, I'm not really familiar with like phototherapy. I was just curious what that looks like, or how long, if you have any idea, like how long you would have to sit under the light or something like that to have some beneficial effect.
So it would be similar to how babies are put under the blue light. They're put in this like plexiglass type cube because they fit in there. So it would be different for adults.
I think.
Go ahead.
No, I think it's basically kind of depends on how much like what they were saying, the units of vitamin D that you can get from those sources like the lamp, the phototherapy lamps and such. So it kind of depends on like the levels of amount that you kind of need to get from that. So
to add on to it, I guess, according to our research, we found out that the time that you were asking about varies according to the disease or like the amount of vitamin D that they need to obtain. Yeah.
Are you talking about a different kind of lamp than the traditional sun lamps that are out now? Because I know that I've read that some of those can cause manic episodes in bipolar patients.
Yes, I would say these are like similar to those, but maybe we would look over into all the other risks associated with it.
I guess in a way we were looking at those too, because you did mention the mania. So that's we kind of didn't specifically mention it, but we were also looking into how that would affect both not just the depression but the mania as well. But that's in a way a good point to test out if we do get to conduct this research, is how that affects the mania as well and overall the bipolar disorder.
Did you find any articles that talked about vitamin D being either regular or maybe high in patients with manic symptoms?
Not specifically. We haven't specifically like that we've searched.
We just found basically kind of like correlation of how vitamin D affects the general gray matter volume of the brain. So in specifics, no, we have not.